Very good morning, dear students. The next topic mentioned in our syllabus, unit number 5, nucleic acids, is DNA double helix. So this is one of the major milestone in the history of biochemistry and studying the entire genetic material that is made by two great scientists, Watson and Crick. And this is given the answer for many unsolved questions in the biology in terms of the structural and functional aspects of nucleic acid. So I am sure that you might have studied DNA double helix in your previous semester or in the lower classes, but here we are going to discuss the detailed aspects of the DNA double helix, the biochemistry behind the DNA double helix. So before we are going to discuss the details, we would like to glance through the evolution of DNA double helical model proposed by Watson and Crick. Okay, as an introduction, we should have a discussion about the historical background and the study. We have in the beginning of this unit we have discussed the historical perspective, how DNA has become the genetic material and how the scientific community proved the transferring material from one organism to another organism is DNA. And here there were a lot of studies were conducted in order to propose the DNA has a characteristic helical like structure, and three-dimensional structure is very important for the elucidation of the function. And the study was started by a great contribution by William T. Osprey, a scientist. He was the first person he thought about the 3D structure of DNA. He was the person he identified DNA could have a 3D model in his mind. Okay, and he initiated the X-ray crystallographic studies, and he proposed a concept of 3.4 angstrom repeat. While he started the X-ray crystallography, he could see that there are strands, they are spacing between 3.4 angstrom. Okay, and hence he proposed the concept of 3.4 angstrom repeat. And later, as I told in the beginning, the great scientist, Rosalind Franklin and Maurice Wilkin, Maurice Wilkin and Rosalind Franklin, they have continued Osprey's study, and they were studied in detail about the X-ray crystallography patterns of the DNA, and they were the first people. They got an X-ray diffraction photographs, as you can see here, of the DNA fiber, and as proposed by Osprey, they could also see this 3.4 angstrom spacing between the strands that is present in the DNA and this helical DNA. And they confirmed the 3.4 the 34 angstrom repeat. That they confirmed the 34 angstrom repeat. So that is the another important concept they have proposed. So these are the greatest scientists. So whenever we spoken about the contribution of a scientist, and this scientist Rosalind Franklin, she has got a great role, but unfortunately people never talk about the contribution of Rosalind Franklin. They always highlight Wilkins' contribution, and later when they received the Nobel Prize, she was no more, and she was died in her very young age due to the cancer. So that is why the people now, the world is saluting her contribution for the initial study of DNA that given James Watson and Francis Crick and Wilkin too developed the three-dimensional structure of a double helical structure of DNA. Then next important aspects as the historical perspective, the great scientist called Dr. Erwin Chargaff, he also given some significant studies. Okay, he proposed that the molar ratio of the nitrogenous bases such as adenine to thymine and guanine to cytosine are very close to unity. Okay, the molar ratio of adenine to thymine and guanine to cytosine are very close to a unity, that is 1. Okay, and this concept is known as Chargaff rule of equivalence or Chargaff equivalence rules. Okay, we will discuss in detail once again. Then another continuation with their studies, scientific community believed that the nucleic acids are a monotonous structure. Okay, which are a monotonous structure of repeating tetranucleotides. Okay, they are repeating tetranucleotides, and they called as tetranucleotide hypothesis. Okay, the nucleic acid is made up of repeating tetranucleotide residues, thus it is known as tetranucleotide hypothesis. Then later about the nitrogenous bases and sugar. Okay, the sugar phosphate backbone is very important, and later the great scientist called Linus Pauling. Okay, Carl Linus Pauling, and he studied about the nitrogenous bases and its conformation. Okay, he studied the nitrogenous bases are projected outward from the central core formed by the backbone of sugar and phosphate. Okay, so nitrogenous bases projected outwards. Okay, then the later after so many studies, the greatest study was conducted by the in continuation with Rosalind Franklin's and Maurice Wilkins study, the continuation of this study done by the James W. Watson and Francis Carey Hampton Crick. Okay, James Watson and Francis Crick, and they have proposed the crystallographic structure of the DNA based on 3 main studies. Okay, Watson and Crick, it was a historical milestone in 1952. It has happened. Okay, 1959. Okay, and it has I think 1953. Okay, it has happened by these 3 scientists. Okay, so that is X-ray, diffraction, base pairing and analytical studies. Based on these 3 studies, okay, they proposed a double helical structure of DNA. For that particular study in 1962, they won the prestigious prize with Maurice Wilkins. Okay, here you can see the great milestone that has happened in the discovery of DNA. Where you can see James Watson, he was very young, just he was 20s. Okay, and Francis Crick. Okay, and they could see they have made the stick model of the double helical DNA, what they have studied in the X-ray crystallography, what proposed by Rosalind Franklin and Maurice Wilkins. Now we will discuss what do you mean by Chargaff rule of equivalence before we are going to discuss about the X-ray crystallographic studies. Okay, there are mainly 5 statements in Chargaff rule. As I said, so the total concentration of the purine and total concentration of the pyrimidine in the double helical DNA are equal. Okay, so that is the sum of the purines, that is known as Pu, is equal to that of the sum of pyrimidines. Okay, equal to that of the sum of pyrimidine, that is purine divided by pyrimidine is equal to 1. Okay, that is what is earlier we have discussed. The molar ratio of adenine to thymine and guanine to cytosine are very close to unity, that is 1. Okay, then the second statement is the ratio of adenine to thymine is 1, that is A by T is 1, and the ratio of guanine to cytosine is also 1, G by C is 1. Okay, the bases with 6 amino groups are equal to that of the bases with 6 keto or 6 hydroxyl groups. This is the 4th statement. Okay, and there is a formula that formula is known as A plus T or G plus C, okay, it's equal to 1, okay. Then that is the ratio of A plus T divided or G plus C, that is known as the dissymmetry ratio. When the dissymmetry ratio exceed 1, greater than 1, such a type of DNA is known as the DNA is AT type. When the dissymmetry ratio is less than 1, such a type of DNA is known as a GC type of DNA, okay. So these are the 5 rules that is proposed by Erwin Chargaff, okay. This is known as Chargaff rule of equivalence, okay. Based on this particular rule, the DNA double helix is proposed. Now we are going to discuss what are the salient features of the double helical structures that is proposed by James Watson and Francis Crick. This you have already studied in your lower classes. So the first one, the DNA molecule consists of 2 identical polynucleotide chains, okay. There are 2 helical polynucleotide chain which are coiled around or wrapped around a common axis in the form of a right-handed spiral double helix, okay. That is the first statement. 2 helical nucleotide chains are coiled around or wrapped around a common axis in the form of a right-handed double helix, okay. And these 2 helices are wound in such a way that they produces 2 interchain spacing or grooves, okay. One is known as a major or wide groove. The width is approximately 12 angstrom and there is a depth of 8.5 angstrom. That is a wide groove or major groove. This is very, very important when the DNA is interacting with other molecule like proteins. And a minor or a narrow groove, okay. The width is half, okay, approximately 6 angstrom and the depth is approximately 7.5 angstrom. And these 2 grooves arise because the glycosidic bond of the base pair are not diametrically opposite each other. Yesterday we have discussed the DNA has stabilized 4 different forces. The 2 strong bond, that is phosphodiester bond, that is there, and the glycosidic linkage is also there, okay. The glycosidic linkages are present between sugar and nitrogenous base, okay. They are not diametrically opposite each other, okay. Yesterday we have discussed they will be able to produce rotational isomers, okay, and due to this particular aspects they are not properly in a diametrically opposite direction. That create a space, okay. That is why it is known as the grooves. Then the minor groove contain pyrimidine O2 and purine N3, okay. Pyrimidine O, second position there is an oxygen atom to the O2 and the purine N3 of the base pair, and the major groove is always opposite side of the pair, okay. The minor groove pyrimidine O2 and purine N3 of the base pair, and the major groove is just opposite to this pair, okay. And each groove is lined by potential hydrogen bond donors and acceptor. Yesterday we have discussed how the bond is produced, okay. Hydrogen bond is produced, again we will see. Then the major groove is more accessible to proteins interacting with the specific sequence of DNA. I told you the major groove has a wide width and depth. There is lot of space in which the protein can come and bind. Thus, this major groove is more accessible to the interaction with the protein with the specific sequence of a DNA. And the 2 helices wind along the molecule parallel to the phosphodiester backbone, okay. And these strands are known as, these strands sometimes called as plectonemic coils, okay. The plectonemic coils, that is the coils which are interlocked about the same axis. They are interlocked about the same axis, okay. The 2 chains run in opposite direction. This is very, very important. There are 2 chain Then one is oriented to the 3 dash to 5 dash direction, and another one is oriented to the 5 dash to 3 dash direction. Okay, they are anti-parallel, which means that one strand, the ring oxygen of the sugar moiety faces upward, while the other strand, the ring oxygen faces downwards. Okay, so these are the two important important aspects. Okay, then the next one is the phosphate and deoxyribose units are found on the periphery of the helix. The next feature is the phosphate and deoxy units are found on the periphery of the helix, whereas the purine and pyrimidine base occurs at the center. Okay, and the plane of the bases are perpendicular to the helical angle. Okay, the plane of the bases are perpendicular to the helical angle, and the plane of the sugars are almost right angles to those of the base. Okay, the plane of the sugars are almost right angle to those of base. Okay, and now this is very very important. The diameter of the helix is 20 angstrom. Okay, and the bases are 3.4 angstrom apart along the helix axis and are rotated by a rotation of 36 degree. Their rotation is 36 degree. Okay, and one helical structure consists of 10 residues on each chain at an interval of 34 angstrom. We have discussed this, both the concept, 34 angstrom repeat, 3 angstrom repeat. So each turn of the helix contain 10 nucleotide residues. Okay, these are the important aspects. Then the two chains are held together by hydrogen bonds. We have already discussed, held by the bases. Okay, nitrogenous bases, and the adenine always pair with the thymine by 2 hydrogen bonds, and guanine always with the cytosine by 3 hydrogen bonds. Okay, this specific positioning of the bases are known as complementarity. The complementarity. Okay, and this is very very important. Yesterday we have discussed the DNA is stabilized by 4 different interactions. Okay, number 1, phosphodiester bond, the interaction between the sugar and phosphate. Number 2, glycosidic linkage, the interaction between the sugar and nitrogenous base. Okay, then the other 2 are weak interaction. The interaction between nitrogenous bases are known as hydrogen bond, and the interaction between the aromatic ring of one base pair to the next base pair below the the strand, that is known as a base stacking. Okay, and the stability of DNA is mainly depending on the van der Waals interaction between the plane of stacked bases. The most important parameter for the stability is the base stacking. Okay, and the sequence of bases along a polynucleotide chain is not restricted in any way. Okay, and the presence of the sequence of the bases that carries the genetic information. So these are the salient features of a DNA. So what I have been telling, you can see in this diagram, the DNA has a long polynucleotide chain, one oriented to this particular direction, that is 5 dash to 3 dash direction, and another one is a 5 dash to 3 dash direction. Okay, they are anti-parallel each other, one oriented this direction, another oriented this direction. Okay, then there is something called a space. Okay, this is known as a major groove. There is a lot of space, and there is here you can see minor groove. Okay, and one turn, this is, I told you that the total length is that this is one turn. The total number of this base pair present in one turn is 10 nucleotide base pair, correctly 10.5 base pairs. That is, one particular turn has a 3.4 nanometer or 34 angstrom in diameter. Okay, and this is the approximately 1 nanometer from the axis. Okay, and the distance between two adjacent bases is 0.34 nanometer or 3.4 angstrom. Okay, so this color is indicating, red is indicating adenine, this yellow is indicating thymine, green is indicating guanine and blue is indicating cytosine. And this you can see the width, okay, the diameter of the DNA is 2 nanometer or 20 angstrom, okay. This is the axis of the helix, okay. So this is, I told you, this is 5' to 3' direction, okay. This is orienting like this. This bond is known as the phosphodiester bond, okay. The bond, this is the one particular chain, okay. This is phosphate, this is sugar, and this is nitrogenous bases, okay. And this other is oriented to, one is oriented to 3' 5' direction this way, other is oriented to this particular direction, okay. And the distance, this particular, this is in the 5' and there is a carboxylic acid group, there is the 3' and there is a hydroxyl group, okay. This here you can see, this is the C, okay. This is the, this is again this carbon atom, okay. So here in the backbone, that is phosphate. So the negative charged, the charge of the DNA is negative, and the bond between sugar and nitrogenous base is a glycosidic linkage, and the bond between nitrogenous bases, that is hydrogen bond, and the bond between the nitrogenous bases, okay. This is due to the pyrimidine nitrogen that is present in the detailed structure, that is known as a base stacking, okay. So these are the important aspects of double helical structure of DNA. Then two important aspects of double helical structure of DNA. One is antiparallelity of the polynucleotide chain. Next one is the base complementarity of polynucleotide chain. One is antiparallelity, next one is base complementarity. So what is this? We have already discussed. The two chains are complementary to each other. They do not run in the same direction with respect to their internucleotide linkage, but there are anti-parallel, okay. So that is one strand is run at 5' to 3' direction, and the other strand is run at 3' to 5' direction. This is known as the anti-parallelity of the polynucleotide chain. Then the base complementarity. This is an important feature of the DNA double helix with respect to their specificity, and there is a specific base pairing is there. The specificity of the pairing, okay. The pairing always occur. Purine always pair with the pyrimidine, or pyrimidine always pair with the purine. So that is adenine always pair with the thymine, and guanine always pair with the cytosine, okay. That means to say adenine, that is known as a 6-aminopurine will always bond with the 6-ketopyrimidine, and 6-ketopurines are always paired with the 6-aminopyrimidine, okay. And the two helix of the DNA molecules are complementary to each other. They are not identical. That is known as base complementarity, okay. Base complementarity. And the base complementarity, the base pairing is mainly due to two important parameters. One is steric factor, another one is hydrogen bonding parameter. We will discuss that, okay. This is the base complementarity. This is the DNA made up of two strands. Here you can see the complementarity of the bases. This adenine always paired with the thymine by 2 hydrogen bond, and cytosine always paired with guanine with a 3 hydrogen bond, and guanine always paired with cytosine 3 hydrogen bond, thymine always paired with adenine with a 2 hydrogen bond, okay. This we have discussed yesterday also. This is the base complementarity. Now what is the reason for base pairing? Why this adenine is always paired with thymine, or why there is no pairing between adenine or guanine, or adenine or cytosine, or guanine or thymine, or guanine or adenine, okay. There are two important reasons. One is known as steric factor, okay. Then the glycosidic bond, that are attached to the base pair are always 10.85 angstrom apart, okay. So this distance, this rung, you can see this rung is 10.85 angstrom, okay. So 10.85 angstrom, okay. A purine pyrimidine base pair fit perfectly in this space, okay. In this particular space a purine You know that purine is a bigger sized molecule. It has two heterocyclic ring and pyrimidine ring and an imidazole ring. Okay, and pyrimidine has only one heterocyclic molecule, 6-membered ring. Within this particular space, this is perfectly fit and they create hydrogen bonding. Okay, then what will happen if there are two purine? If there are two purine, okay, there will not be insufficient space. There is insufficient space for a two purine in this 10.85 angstrom. If two purine means, you imagine this is one, and if I keep here, okay, then here 2 and again this is here, okay, then 1 2 3 4, that is practically not possible within this 10.85 angstrom. That will not fit. Okay, so that is why there is no adenine guanine bond. Okay, so that is the reason. Then you will ask, then why can't these two things together? Okay, then what will happen? So whereas there is more than enough space for two pyrimidine, so that they would be far apart from the hydrogen bond. Okay, so if you keep this is here, then what will happen? This is a single bond, single six-membered structure. If I keep it here, then what will happen? This, if I keep it here, then you can see that this will be one only single bond and one more single, this here it will be 6-membered ring, one single 6-membered ring, another 6-membered ring. There will be one is here, another one is here. There will be lot of spaces between these two. There will not be having a close proximity to produce the hydrogen bond. Hydrogen bond will not produce because that is very loose actually. Okay, that is why there is thiamine will not pair with the cytosine. Okay, thiamine will not pair with the cytosine. So that is what is the steric factor. Okay, so that is known as steric factor. So one member of the base in the DNA is always should be purine and other should be complementarity with the pyrimidine because of the steric reason, because there will be steric clashes and there will be the problem with the arrangement. So that is known as the steric parameter. Then what about hydrogen bond? Okay, the hydrogen bond atom in purine and pyrimidine bases have well defined position. Okay, in the case of hydrogen bond atom, in the purine and pyrimidine bases are in a well defined position, because adenine cannot pair with cytosine because they would be two hydrogen atom near one of the bonding position at the other. Okay, and none at the other. Similarly, guanine cannot pair with the thiamine. Okay, I will tell you what is the reason. And adenine forms 2 hydrogen bond with the thiamine, whereas guanine formed 3 hydrogen bond with the cytosine. Thus, the GC bond is very, very stronger than by 50% than the AT bond. Okay, and the higher the GC content of the DNA molecule, the density of the DNA, the buoyant density of the DNA will be very, very high. If the GC content is high, the density also will be high. Okay, so that is the important thing. So as you can see here, why there is the hydrogen bond should be like this. Okay, so thiamine, this yesterday we have discussed. Imagine this thiamine if I am keeping down. Okay, then what will happen? Guanine paired with thiamine, then what will happen? Here you can see, if it is here, then you can see in the case of guanine there are in the case of thiamine there is NH. At the same time you see here also H. Okay, here it will be NH and NH. Okay, this between 2H there will not be any interaction. Then here you can see, here it will be N. Okay, and this one you see again NH, again NH and NH. Okay, so NH and NH there will not be having hydrogen bond formation. Okay, 2NH if it comes, it will not be having hydrogen bond formation. So it will be always NH or NHN or OHN. Okay, it will not be like NH HN. So that is the reason. Okay, so that is the important thing. Okay, there will be the hydrogen bonds will not be, the hydrogen will not be coming into the close proximity to produce the bond. Okay, that is another important. These are known as the hydrogen bonding parameter. Okay, so this is all about the salient features of a DNA double helix. So dear students, we will conclude. Okay, So today we have discussed the evolution of a DNA double helix. Okay, initially we have discussed the historical background. Okay, the evolution. Okay, the evolution of DNA double helical structure. I'm sorry, there is a repetition. Evolution of DNA double helix. Okay, a historical background. Then we have discussed Chargaff rule of equivalence. Okay, we have discussed about the Chargaff rule of equivalence. Then we have discussed the features of the helical structure of DNA, the Watson and Crick model of DNA. The next one is antiparallelity of the polynucleotide chain. Then we have discussed the base complementarity of polynucleotide chain. Then why there is a reason for base pairing. What is steric factor? What is hydrogen bonding parameter? Okay, why there is no bond formation between two purine, or why there is no bond formation between two pyrimidine, why always one purine and one pyrimidine? Okay, we have discussed due to this particular thing. So this is all about this particular topic, dear student. So these are the textbook. So Principles of Biochemistry by Leninger, as I said, always you can refer. Then similarly, any of the books are very good. Okay, then I specifically refer this Biochemistry by J.L. Jain. It's very simple, very detailed it is written. Okay, this you will be able to understand. So thank you very much for watching this particular class. If you are having any clarification, you can ask me. Have a great